there is one in Forsmark, but they also have a heated bay. There is actually also, I found a study also in China and England, and that around the world they're using similar system but other designs. There was some study they put down concrete and just let biofilm grow, where we used the natural material, the granite stone that is naturally there.
Mm-hmm. And when you sample the biofilm on the stones,
Yes.
do you, well, later on you look at your sequences that you get from that, but do you have any idea how this biofilm looks like? I mean, visually, did you somehow visualize the biofilm? I mean, how it is arranged, how thick it is, which color it has, whether it's, you see a layer of algae or something like that. Did you also make notes, or did you look in a microscope or whatever? I mean, to get an idea how does it look like, the biofilm on the stones?
Well, I was there every time we took biofilm. We didn't note down. We just scraped the biofilm, and sometimes it seemed like the stone is clean, but there is still something there.
Yeah.
So sometimes, especially in the heated bay, it was like you can see it very easily. It's all green matte, as in the picture I showed. And sometimes in the other environment, it's like just sitting there scraping a stone, and it looks like a stone.
Yeah, okay. But you didn't look in the microscope for what you scraped away from the stones?
No, it was like DNA was extracted and so on. We didn't look before.
Yeah. And yeah, you probably also realized that this biofilm is in a matrix, embedded in a matrix.
It's many organisms. So a biofilm form normally that you put like some kind of hard material. That can also be a ship. Unfortunately, they also do that, biofouling, into the water. And the first that will happen is that bacteria will attach, and when the bacteria have attached, other organisms attach on them. So they form this film made of many different organisms. And when the biofilm get too big, too thick, then like they are budding off and in the water column, and then they found a new surface to colonize.
So a kind of succession, no, of the biofilm.
Yeah,
But I also was wondering about the variability, no, in the diversity data. No? When you have one stone and another stone and another stone, and then you analyze the biofilm in terms of diversity, and then you compare it with another system, and probably every stone is a little bit different, I can imagine.
There is patchiness, of course. There is a patchiness. It is biofilm. It responds a lot to environments. But still, there is more similarity within an environment than between an environment. So we argue that we can take just one stone. Something I thought about after was, because when we did this fieldwork, what took a really, really long time was to walk through the jungle, because there's not even proper roads in many of the places we were, and it would take a long time is to sterilize everything so we don't have contamination. But we could maybe have scraped 5 stones and put it into one tube, so one sample will be 5 stones, but still it is, we took just one, and it is like we were there multiple times, so still we have good data.
But you sampled several stones, I guess, for each sampling day, not only one.
We had spots during the coastline in the tree environment, and every time we were there, we took a stone and we scraped it. So we had like it was 10 in the gradient and 8 in each of the tube bays. So we have multiple samples, but from spots along the coastline.
And one spot means one stone.
Yeah.
Means not one stone, but one little area from the stone.
Predefined area.
Yeah. Did you ever check when you sampled different areas from one stone, how different they are, how variable they are?
That's something we should have done,
from
actually.
the same stone?
Yeah, that's actually a good idea.
Because then you would have replicated at least from the same stone, because there's also already some variability, I can imagine, on the same stone, depending whether you sample from the exposed more to the other side and yeah, and then, well, it looks from these stacked bar plots look as if there's quite some variability between the different sampling sites. And I'm wondering, I mean, then you compare the two bays with the different sampling sites. And I mean, we had another study in the Baltic where we had also a problem where we wanted to compare different areas, and we have a high variability also in bacterial composition. And now we are thinking more, I mean, how many samples do you need with a certain variability to distinguish these areas? Did you ever think about that? I mean, how many samples do you need with a given variability to be able to detect differences? You can say, I want to detect 10% difference, or I want to, for me, it's enough if I detect 50% of a difference between the system. But you need a certain number of samples because of the variability. Did you ever make these considerations or these calculations about how many replicates you need to, or you just did it and then you did the test and saw, well, we see a significant difference?
Well, we talked about how many. Also, we have to take all in the same day, and it takes a long time to go between the spots because we're like crawling up mountains and jumping over trees, and it's really, and we have to carry this big container to put the stone on ice when we were doing the translocations. So it's like logistically really challenging already. But now when we know there is seasonal variation, there is difference between each of the environment, then we could make a longer and more complicated study. But now we know there is differences.
But on the other hand, I have the feeling when I look at your Shannon diversity, alpha diversity, your plots, also these differences are not huge, no? Sometimes they're relatively small, the differences. And you say that when you go to the heated bay, you see a green biofilm, and the other one might be clear stone. So maybe there are other parameters that give you more information on the differences, no? I mean, other parameters than the sequences itself, because the sequences, they show, well, you calculate the alpha diversity and it's very similar. But if you say, well, for example, I measure the thickness of the biofilm or the color of the biofilm or whatever, maybe there are other parameters that show that these biofilms are very different between the heated bay and the control bay, no?
That's something we could do in the future, because there was someone more green, someone more brown.
Yes.
And some of them you could just feel there was something slimy on the stone, but you couldn't really see it.
You didn't measure this EPS structure in the matrix, for example, this polysaccharide. I mean, it's possible, I think, to measure it also, and the amount of. So maybe there are other much more pronounced differences between the two sides for other parameters, no, to check. A more technical thing. I have never used this mixoplankton database to distinguish. Is it reliable? Do you think? Would you recommend this database to distinguish heterotrophic and mixotrophic taxa? Is this solid?
Well, the data is only as solid as the database, and of course, not all species have been investigated if they are heterotroph, mixotroph, phototroph. So, over time, when people do more and more investigation, it's going to be more and more complete. But as for now, this is what we can do.
And at what taxonomic level, or what taxonomic level is required for this database? I mean, I can imagine that a lot of your sequences, your 18S sequences, are annotated to a certain taxonomic rank, but not always to the species level, right?
No.
Maybe sometimes it says unclassified, unclassified straminopile, for example. So what do you do with these sequences that are not classified to the species level? Do you also put them into the Mixoplankton database? Do you use it for these sequences as well?
Well, if there is no data on that level, then we can't say anything about it because the database is only as good as it is.
Yeah.
And we couldn't investigate all the species ourselves.
No, of course. And that would be my question. How to handle such data? And I mean, also, when you take, for example, the chrysophytes as an example, Chrysophycea as a family, right? I think you, in your table, I don't remember exactly whether you judged it as a mixotroph or as a phototroph. But in fact, in this family, Chrysophycea, we have heterotrophs, mixotrophs, phototrophs. We have all three nutritional types. And that means if I do not really know which taxa it is on at least the genus level, I would say, I cannot give the sequence to this Mixoplankton database, right? That my question is a little bit whether you filtered your sequences before you use this Mixoplankton database. For example, taking only out the sequences that have been assigned a genus level or something like that.
We just have to accept that sometimes it's unknown because it's not classified down enough that we can actually know.
Yeah, but hopefully the Mixoplankton database is not assigning a certain term to these unclassified sequences and says, well, this is, I don't know, straminopiles, and we say they are mixotroph, but because it can be everything. Yeah? And then I have a question. You mentioned these hypotheses. You expected that on the biofilms that the micro-eukaryotes will show less differences between the base and a weaker response than the prokaryotes.
Yes, there was differences between the two domains, and and you also have to think about that prokaryotes in general have a faster generation time, meaning that they have a higher responsiveness to environment. So they respond more to changes. Did I explain?
They are faster. Yeah, I believe they are faster, but the bay was heated for 50 years, so that should be sufficient time even for higher organisms to respond. No.
Yeah.
So why do you expect less response for the micro-eukaryotes and for the prokaryotes in this comparison? I mean, there was sufficient time, huh? Even that they are a little bit slower, but there was enough time, huh, to respond.
Yeah. Well, we also see, in general, we see that there is a more responsiveness to environment in the prokaryotes compared with the micro-eukaryotes.
And wait, what I want to say. And I mean that at the end you confirmed this hypothesis. No? There were less differences for micro-eukaryotes than for prokaryotes, right?
Yeah.
I was wondering, because in this plot you showed, you didn't specify at which taxonomic level this comparison was done. Was it the same for prokaryotes as for eukaryotes on the same taxonomic?
Yeah, the same taxa.
And on which taxonomic level was this comparison done?
Oh, I think it was genus.
ASVs maybe?
Yeah, we used ASVs.
For bacteria and for eukaryotes also? For both?
We have the ASVs, yeah.
Ah, okay. So because not because that would be a problem if you would use different taxonomic levels. Let's say here we use ASVs and here we use genera or something like that. Then it's not directly comparable. But also ASVs for eukaryotes, yeah.
We use the ASVs.
And do you think ASVs is a good representation of species?
Yeah, it's a very good proxy for biodiversity because the ASV in the RNA it responds to a specific sequence, and if in this sequence there are one just nucleotide different, then it's another one. So it's actually a really good method.
But can you also imagine? Do you think it also can overestimate or underestimate the real diversity ASVs?
It could be that two different taxa just have the same nucleotide sequence, but it's still a really good proxy.
Yes. And I also heard that some taxa can have different ASVs, so even within one taxa, because they have different copies of the RNA, and that it can get, then you get an overestimation of diversity with ASVs. And then you had these biofilms, natural biofilms on stones, but then I read that you also had looked for the biofilms on these homologas.
Homologas, yeah.
Homologas. So this is a different surface than a stone. It's a metal surface, I guess.
No, it's plastic.
Plastic surface. Okay. Are there systematic differences in the biofilms between on the plastic surface and on the stone surface?
The thing is, we cannot really disentangle the effect of substrate from the effect of depth, because these homologas were attached to the cages we put much lower, because we had the cages with the sediment, and on these we had homologas to take temperature in the deep, and this the biofilm is like the water was up to my knee, you know. So there is both an effect of how deep was the water and how was the substrate type, because of course the substrate type have a lot to say. If it's on a natural, even between stones, there can be differences, and how will the biofilm actually form and grow.
Yeah, you would have to put a homologa near a stone in the shallow water,
and a stone in the deep. Yes.
Or a stone in the deep, and then to make the comparison, probably. Yeah.
Yeah.
Yeah, and then, but maybe this is we already discussed that when you come with a conclusion. For example, regarding community composition, temperature had a weak but significant effect on microeukaryotes and a marginally non-significant effect on prokaryotes. I'm always a little bit skeptical when you say, well, temperature had an effect. You mean the heated bay is different, but and then temperature might be the reason. But then you have the other factors, no?
Yeah,
keep that in mind.
We have many other factors also, but we were focusing more on the temperature. It could, of course, be that there is a difference, but it was not only by temperature, because we are in a natural environment. It's like nature. There are so many factors in this system, but it's a really good study system because we are in nature. Everything is natural.
Yeah, I understand. But then I would prefer to read that, well, the heated bay has a significant different microeukaryotic community, for example. But when you say temperature had a significant effect, you do not really know whether it's really the temperature or it's a higher productivity of the system or the light, the different light climate. So it's a range of factors, no, that might be responsible, and temperature is one.
Yeah.
One of them.
Yeah, that should have been reframed.
But not the only one.
Yeah, of course.
Okay. Yeah, and one factor, I was also thinking of one factor. I mean, we talked about light, nutrients, productivity, but maybe also the food web could have an effect, right?
Yeah, because we see that other grazers between the two bays.
Right.
So maybe other species are also different. We haven't looked at snails, all that.
Yeah, exactly. Do you know something about the trophic interactions going on in biofilms?
What is the question?
Trophic interactions between different organisms happening in a biofilm? Like in plankton, you have fish eats the copepods eats the phytoplankton.
Yeah,
These are the
food web.
trophic interactions. But do we have similar or different trophic interactions on the biofilms?
But biofilms is on the absolute bottom of the food web, and they have different grazers in the two bays.
Well, but still you can have trophic interactions in a biofilm, no? If you have micro eukaryotes and bacteria.
Yes.
Right?
Mm-hmm.
This is at least one trophic interaction I can imagine of.
Oh yeah,
no?
they have.
How do they live, the micro eukaryotes? What do you think? What they are doing in the biofilm?
So the microeukaryotes, if they're phototrophic, they take the sunlight. If they're heterotrophic, then they feed on carbon sources like humans are doing, they eat. And if they're mixotroph, then they can switch between. So if one food feeding is favorable, they just change between them.
Mm-hmm. So if they are heterotroph, they
eat carbon, they consume carbon, and there's
carbon in which form? I mean, carbon pellets, or what kind of carbon do they consume?
Just food.
What?
Just carbon sources for food.
Yeah, but from where do they get the carbon? Is it an organism, or is it dead carbon, or what?
They can probably eat both living and dead.
Yeah. And living carbon they get.
What?
Well, I was thinking of bacteria. Maybe they graze on the bacteria in the biofilm, no? Or the prokaryotes.
The microeukaryotes eats the prokaryotes.
Yes.
Maybe they do.
Yeah, I mean, what else? What else should they? I mean, the exopolysaccharide matrix maybe, but I don't think they can eat that. So they should try, yeah. But I thought that there's also the general picture, or it has been in the literature, that biofilms are protected due to the matrix. They are protected against grazers, against microeukaryotic grazers. I don't know. Do you know something about this? But like whether there's really grazing going on in the biofilm?
Well, I only know what I've heard about the other study, that there are different species of grazers in the two bays. I have not studied specifically grazing.
Yeah, but probably there's grazing ongoing. Huh? Same with the metazoans. Okay. I don't know whether you know you showed these stacked bar plots on the class level, I think, for the prokaryotes. Was it on the class level?
I don't remember.
Or on the phylum level, I don't know. But I was just wondering whether you know some of the dominant taxa. Yeah, that we can, some organisms, to have some organism names that are dominant in the biofilms, maybe in the heated bay and in the control bay. Which taxa dominate in the biofilm?
Cyanobacteria. Cyanobacteria. There all the time.
Okay. Can you go with cyanobacteria a little bit down? I mean, specify which type of cyanobacteria these are?
The names are in the supplementary materials.
Yeah. And for the heterotrophs, do you also have groups for the heterotrophic that dominate?
I don't remember.
Not really. Or for the protists, for the microeukaryotes, which microeukaryotes are dominant or are so important on the biofilms? Do you remember some?
I don't remember the names.
No, okay. Good, then let me see. With the translocation, with the translocation experiment, I was wondering about one, when I heard your talk, one question came to my mind, another one. You studied how the community changed when you translocated it, but from the control to the heated or vice versa, how they became more similar to the recipient community, right?
But there was still legacy effects.
Still legacy effects, a little bit different. And I was thinking, what are the underlying reason? What is the underlying reason why they are not becoming?
Because they formed in the opposite environment and they are still, for some reason, shaped by that. But one thing we could do in the future was to have a longer incubation time, because it could be that we just harvest them too early. We don't know. Actually, what would be really good was to introduce even more and translocate even more, and then have like after half a year, 1 year, 1.5, 2 years, like have, so we can see the more of a full picture. Because what we can conclude is like, oh, that was not a fully conversion. Yeah, but maybe in 2 years that would have been, and it's something we actually don't know.
That could be one explanation. And I was thinking of another explanation possible, because with the sediment cores, you do not transfer only the community to the new system. You also transfer the sediment itself with everything which is in the sediment, right?
Yes.
And then you think of the environmental factors, temperature that is affecting it, but you bring new substrate and nutrients and contaminants, about everything which is in the sediment you also bring to the new system, and this is not exchanged, right?
Do you mean we should have taken just the prokaryote out of the sediment? And
that's not possible, but you have to consider that, no? Or maybe a sterilized sediment or whatever. I don't know. I have no solution for it, but I was thinking that it cannot be similar. It cannot be 100% similar to the recipient system, because you still have the sediment from your other system with everything inside. And this is not similar to the recipient system. And that I would expect that there's always a difference, maybe even after 2 years that there's a difference, because some components in the sediment might be different. But we don't know in detail which components in the sediment, some of the nutrients, some of the porosity of the sediment structure and things like that. I was thinking that could be a principal reason why they cannot become totally similar, or maybe after a very long time. But
let's see.
Okay. Well, maybe we can end up with that you say something about what puzzles you most from this study, or where do you see still gaps in our understanding? What is the open question, and where you think, well, I would have needed more time to study that, or new techniques, or whatever. Where do you see what is your major gap in your understanding in this system?
So you ask me if I have ideas to like extend studies, what my ideas are?
Yeah, not. I guess you have many ideas, but maybe all during your 4 years of study, there was always a point where you thought, well, I'm not sure about this. How can I approach this? Is there some uncertainty or an open question, or there's something you do not know yet?
Yeah, I would like to do more translocations, because we only introduce and translocate, but it could be pretty interesting to like put it back and see, okay, now it was there first, then here, would it like be like, oh yeah, I'll go back to my first state? Or would it be like, no, I want to also have legacy effect from that environment. And also to have more, okay, we only have 2 bays, but we could have had more look at other environments and translocate between to have more different temperatures and more different environmental factors.
Yeah, that could be interesting. Okay, I think, well, I have all my questions, most of them asked. We have discussed that. I think from my side, I am fine, and I give the word back to the chairman. Thank you very much.
Okay, thank you, thank you.
Thank you very much for a nice discussion. Now we give the word to the examination committee, and maybe we start with Professor Eva Lindström online.
Oh.
Okay, can you hear me well?
Hear you loud and clear.
Good. Yeah, thank you for a very nice thesis and a very nice discussion. I really enjoyed listening to you. But I have a few questions, and the first one is just for curiosity. So you were looking at the two bays, and the one was heated, and it was in the water. The water was heated, but you were also looking at the sediments and going in a gradient deeper and deeper into the sediments. And I wonder, do you know how much the sediments are actually heated and how far the temperature is changing down in the sediment?
We didn't put homologers into the sediment.
So do you know if anyone has looked at it to see how much is the sediment actually heated? I understand that the surface sediment will be heated, but how far will it translate? Nobody knows. Or no, okay. I was just curious to know, but maybe nobody looked at it. And the other question I had, so you talked about the terminology and you talked about the microeukaryotes, and I was a bit curious about, you used the term prokaryotes quite a lot, but I don't think you studied the archaea.
Yeah, they were also included.
They were included?
Yeah, okay. But the primer we've used is more good for the bacteria than the archaea, but it's always a trade-off when you choose a primer.
Yeah. Yeah, okay. So you think that you didn't get a good picture of the archaea as good as for the bacteria? Is this what you mean?
The sound is a bit weird.
Okay, sorry.
Can you ask them?
Yeah, so my question then is that you think that your picture of the bacteria was better than the picture of the archaea?
Yeah, probably.
Yeah. Okay, then I understand why you used the word prokaryotes, because in the discussion now, I almost felt that you used the word prokaryotes and bacteria equally, and that I would object against. Oh. Yeah. Yeah, okay, nice. I had a question about some figures. You had a type of figures I haven't seen before. They are called the heat trees.
Yes.
For example, in paper 3, you show these heat trees in figures 5 and 6. And I wasn't sure how to interpret those figures. So maybe you could explain a little bit?
Yes, of course. So when we look at, if you look at the one that is all gray, so in my thesis, it will be number C. You can see that there is a full conversion. So you take the ASV that is from, when you take from the control and put it into the heated, and you compare it to what is resident in the heated bay, and there is no difference, meaning a full conversion. That's why the tree is all gray. There is no colored areas. So if you look at, for example, figure F, you can see that what is taken from the heated bay and put into the control bay and compared with what is resident in the control bay, and the area that, the more area that is colored, the more differences that is between the two that you are comparing.
Oh, nice. Then I understand much better. Now they are so small, so I can't see what's written in the figure. But so are the dots? Are they different ASVs? The sort of tips of the tree, is that different ASVs, or what is that?
Yeah. So what is colored is what is different between the two you're comparing. The ASVs.
Yeah. Okay, it's ASVs. And is this sort of tree structure, is that phylogenetic? The structure of the tree, is that a phylogenetic tree, or how is the tree constructed?
I'm not really sure about the shape of the tree.
Okay. All right. Oh, I was just curious. It looks like a very nice way to present these results. So that's why I wanted to have some more information. But I think I'm done with my questions. I leave the word to the rest of the committee.
Okay.
Thank you very much, Eva. And then I give the microphone to Johanna Sjöstedt, docent from Halmstad University.
Thank you for a very nice presentation and discussion. There is one general thing.
One what?
One general thing that I'm a little bit missing in this thesis.
Okay.
And it's about functional performance. Mm-hmm. So you have studied how the composition is affected, mm-hmm. but not so much if it affects the function.
Yeah.
So in paper 2, you actually mention functional stability. Mm-hmm. And you say that the functional stability is greatest in the heated bay. Mm-hmm. But I don't understand what this is based on.
So we didn't take metagenomic or anything, but we can still see that in the alpha diversity, when you do the translocation, it seemed like the translocation do not really matter, but for the species composition, it do matter. Meaning that when the species change, they might have had the same function as another species. That's why we don't see in the alpha diversity that there really is a difference.
Yeah, but can you really say that from only studying the diversity and the community composition? Don't you need to measure some kind of function?
Well, we can speculate, yeah. We should probably have taken metagenomic, but there's something that can be done in the future.
Yeah, that would be a way to actually investigate it. Mm-hmm. Because I think also you mentioned in your introduction to your thesis that even if there is a change in diversity and community composition, there could still be functional redundancy.
Yeah, meaning functional redundancy, meaning that you change, but you have the same function.
Yeah, that would be an interesting way to continue these studies, I think. I also have a question for paper 3, which I think is a very complicated setup.
Yes.
Yes. So this is actually also related to these heat trees that Eva asked about.
Yeah.
Because I don't really understand which stones that are compared to each other in that heat tree.
Yes.
Because when I read the figure legend, it seemed like you have introduced stones in both bays, both in February and in April 2022. And you have also introduced stones that were later translocated at the same time points, but they were then incubated for 3 months before they were translocated.
Yeah, so we put everything in and then we translocated some of it. So it have different translocation, it have different incubation timeline, of course.
Yes, but doesn't that complicate the comparison? Because you have shown that both the harvesting time and the introduction time point matters for the community composition.
Yeah, actually it do. Yeah.
So so what I'm thinking about is, did you compare the stones that were introduced in February and then later translocated 3 months later to the ones that were also introduced in February?
Yes.
How did you compare it?
So everything we translocated, we put in, and then each season we translocated, and then they stayed for 1 year, and then we harvested. But the resident stones, we had many resident stones, so they were introduced, and then they stayed, and then the months, so they were introduced, and then the months where we harvest them should be the same where we harvested.
So the harvesting month is the same for those that are introduced and those that are translocated?
Yeah.
Yeah, okay. And then just one final question. What would be the next experiment that you would do if you would continue with this study system?
Yeah, I would like to do much more translocations because I think it's quite cool that even you put it in another environment, it still have a legacy effect. So it's still affected by where it was before. And it could be really interesting to just keep translocating to see, okay, if I put you in between different environments and if I translocate you multiple times into the same environment, would you go back to the environment you know? Or will you be like, no, it was actually better to be like the other environment? So a lot of that could be done.
Yeah, thank you.
Yes, thank you, Ida. It was really a pleasure to read through your work, and I feel that I really learned a lot, especially on this biofilm organisms and the biodiversity and responses. I have a bunch of questions, so I will choose a few of those. But I have first a question of the title of your thesis.
Of the what?
The title of your thesis. Are you fully happy with it?
Okay.
Are you happy with your title?
I think it's long.
Sorry.
I think it's long,
yeah.
But it's a really good tell what I have been doing.
Yeah, it's quite specific. You have the climate change there, and you have something about the experimental approach, and you have the target, and you have the place. But I was thinking of these lower trophic level organisms.
Yeah.
Have you only studied lower trophic level organisms?
Well, the biofilm is on the absolute bottom and also the prokaryotes in the sediment, but the copepod is one level above.
Yes, I was thinking that when you study biofilms that also contain a lot of decomposers. And in a way, you can think of the decomposers being the highest level in a trophic structure, because even the top predators die sometimes, and they are degraded and decomposed by some of these organisms you are studying. So okay. I would have preferred maybe primary producers and decomposers in the title.
Mm-hmm.
Then I have a question about the first paper, and the opponent already took the same issue, saying that his colleagues don't believe in your idea that there has been some sort of microevolutionary change in the life history strategies of copepods.
Can you say again?
Your opponent told that some people don't believe in your idea of life history changes happening in copepods between these heated and control bays.
Yeah, we can't really disentangle like microevolution from plasticity, so that's how it is.
But what is your own idea? Do you think that some sort of evolutionary change has taken place in those populations living in these two different bays?
Yeah, so chances over time, I believe that it had happened that they go and make this egg because it's over generation. When you live in an environment that is always like a really good environment, like time-wise for the eggs to hatch, then why should you produce a dormant egg when you can just produce eggs that are immediately hatching? So I think there's like some kind of evolution going on. So over time, they have been more towards an immediately hatching egg. And maybe if you look at it again in 10 years, maybe they have only immediately hatching egg.
If you think of some sort of evolutionary change taking place between these bays or populations inhabiting these two bays, What would be the preconditions for such an evolution to take place? What is needed for an evolutionary change taking place between generations?
In the gene, that there is a difference in the nucleotide, so that the DNA will change and there will be another gene expressed. Yeah, is that what you mean?
Yes, there needs to be selection taking place. There needs to be heritable variation for which the selection can operate on.
Yeah.
Then it often happens that if there is a local selecting for some sort of trait, then this local selection may be kind of counteracted by gene flow from elsewhere. So what is your idea? What kind of gene flow might be taking place between these two bays in copepod populations?
I don't really understand the question.
New
speak
genetic
more into the microphone. I can't hear you.
There may be new genetic alleles coming from elsewhere to these bays, and if that is the case, if there is lots of gene flow, there can't be much of local adaptation taking place.
So you mean other copepods swimming into the bays?
Exactly, yeah.
I think it is unlikely in the heated bay because of the water flow, because there is a constant current like out of the heated bay, so I don't think copepods can go against the currents.
Yeah. How about, can they survive through the nuclear power plant system?
Yeah, it has been measured that there is very, very, very few copepods coming from the water itself. So actually, it's not a problem because there's so few.
In the Baltic Sea, we don't have tide, but we have quite a lot of water level fluctuations that are related to wind-driven currents and related to water pressure differences among different parts of the Baltic Sea. So these water level fluctuations will transport huge water masses along long distances in the Baltic Sea. So I would guess that copepods that are kind of sticked into the water column, they move with these water currents, and therefore they can move from one bay to another, or from the pelagic areas to the bays, or vice versa. Yeah,
of course, if they are in the water that is inflowing, I haven't really thought about that. But of course, then if they are gonna mix with the copepods, then there will be lesser chains. If they are mixing with the copepod that is already in the base and have been infected for a long time.
Exactly. That would, if that is the case, it would make this microevolutionary change less likely.
Yeah.
Okay. Then a question related to, somewhat related to this thing. In the first paper, in the figure 2, that you already showed in your initial lecture, you nicely summarized the differences in abundance of copepods between heated and control bays. But I had an idea, or I understood that you interpret it as like species having different reproductive cycles in these two bays.
So the reproductive cycle, the cycle is that they make the egg either dormant or immediately hatching. It hatches, it becomes a napoli, it becomes a copepod, they make an egg. It's the same.
Yeah, I'm kind of thinking that there were some differences in the species copepod species composition among the bays, wasn't there?
Yeah, it's in the supplementary.
Yeah. So in the warmer bay you had Acartia and Harpacticoids, and in control bays Cyclops and Eurythemora species. I think that those were the main differences.
Yeah, of course, yeah.
So could it be that these differences that you here show, for example, considering total abundance, that they were mainly due to the differences in species composition among the base, but if you would look at certain specific species, would they still be different, these abundances, or would they be more or less the same?
We have a figure in this supplementary where we look at different species, and there are different curves for the different species. I can find